Yours.
Thank you. All right, it's an honor and a pleasure to be here. These are my conflicts of interest. So co-authors on this study are employees of Pacific Biosciences, and they also contributed to genome sequencing data I'm going to show. So over the last 2 decades, the development of arrays, exome, and now genome sequencing has really led to major improvements in our ability to diagnose genetic disease. However, even in the best circumstances, it's very typical that you can only identify causal mutations in a minority of patients. And this suggests that there are alternative mutational mechanisms that are not well assayed by these methodologies. Tandem repeat expansions are one possible explanation for this. There are now known to be more than 50 repeat expansions known to be involved in human disease. Probably the best known of these is the Fragile X repeat. This is a CGG repeat expansion that occurs in the 5' UTR of the FMR1 gene on the X chromosome. In rare cases, this expands to more than 200 copies, and in this case, it becomes highly methylated, resulting in silencing of FMR1. Now, these kind of repeat expansions are very tricky to detect, even with Illumina genome sequencing, and so we hypothesize there's likely to be more of these lurking in our genome that we have not yet discovered. So today I'm going to tell you about a 3-part study where we first tried to identify novel methylated repeat expansions similar to that which causes Fragile X. And then we tested their effects on human traits using phenome-wide association studies, and then investigations in rare disease cohorts. Now, our methodology to find these methylated repeat expansions really came off this prior study from a few years ago, where we observed that using methylation profiles is a really effective way to find these kind of events. And the idea works a bit like this. So we developed this and applied it to this cohort of nearly 6,000 individuals that had Illumina methylation profiles and Illumina genome sequencing. And what we do is we perform an outlier analysis to look for rare gains of methylation in these individuals. So what I'm showing you here is the methylation profiles in all 6,000 individuals at this promoter of this gene, ABCD3. And you can see there are 3 individuals that have rare gains of methylation compared to everybody else. Now what we see is that underlying this gain of methylation is a CGG repeat annotated. So in these circumstances, what we do is then genotype this repeat using Expansion Hunter. And this is the genotype distribution we saw over here on the right. So most people have short alleles of this repeat. But you can see there are a few individuals that have repeat expansions. And the 3 individuals shown in red here are the exact same 3 individuals shown on the left that have the gain of methylation. And because both of these are very rare events, the chance of them co-occurring, the gain of methylation and the expansion in the same people, is incredibly unlikely to happen by chance. With our permutation testing, that was observed less than 1 in 10 million times. And so this is telling us that this gain of methylation is caused by an underlying expansion of this repeat. So we applied this same method genome-wide and identified in total 24 methylated GC-rich repeat expansions. These are shown here. Of these, 7 have already been associated with human disease. And so we hypothesize that the remainder therefore represent excellent candidates for novel human disorders. Before proceeding, we wanted to validate some of these events, so we used long-read genome sequencing. And here, for example, in the FAM193B gene, observed insertion of more than 1,000 copies of this CCG motif in an individual with a gain of methylation. We also looked at their effects on downstream expression using the GTEx project. So we first identified carriers of some of these rare gains of methylation, and then used allele-specific RNA-seq to show that those individuals had unusually low expression and unusually biased allelic expression, suggesting they're causing monoallelic expression. So we now have 28 GC-rich repeat expansions. That's the 24 we found, plus 4 others reported previously in the literature that we know get hypermethylated and cause gene silencing. So really the question is, what are these doing to human phenotypes? So here we turn to the UK Biobank, and we specifically used about 168,000 unrelated Europeans that had Illumina genome sequencing. We directly genotyped these 28 tandem repeats using Expansion Hunter. And then we convert these quantitative repeat genotypes to binary calls of expanded or not expanded. We extracted phenotype data for about 10,500 binary and quantitative traits, and then perform a phenome-wide association study, where we associate the presence of expansion with each one of these traits. And then in total, we found 156 significant trait associations with 17 of these expanded repeats. Now, I don't have time to tell you about all of these, so I'm just going to show you data on 2. This plot here on the left is showing the results for the C9orf72 expansion, and this is already known to be the major cause of ALS. Just to explain, in these PheWAS plots on the x-axis, we assign the 10,500 traits into major phenotypic categories, usually by organ system, and the y-axis is showing the minus log 10 of the FDR. Now, as ALS is a neurodegenerative disease, the major traits we see are indeed reduced brain volumes and altered brain structures. We also see various traits known as features of ALS, such as insomnia, altered frequencies of anxiety, dysphagia, that is an inability to swallow, urinary tract infections, reduced lung function, altered immune function, worse performance on touchpad tests that the UK Biobank uses for some of their surveys. And then we see things like increased frequencies of CT scan of the head and diagnostic imaging of the central nervous system, which are tests commonly ordered by clinicians in ALS. And then finally, increased death rates due to motor neuron disease or respiratory failure, which is the known major cause of mortality in ALS patients. So our results here at this known locus really recapitulate the known symptoms, exams, and effects of ALS and gives us a lot of confidence that our test is working well. And just to give you some insight into the underlying data, here I'm showing you this Phe code for death due to motor neuron disease, and you can see the individuals that were assigned this have a 61-fold enrichment for expansions compared to people that were not. So what are the novel things we found? That's really the more interesting stuff. So the one I'm going to describe to you here is a GCC repeat expansion in the promoter of AFF3. This is a gene located on chromosome 2. Here is our PheWAS results. And the main hits that we saw were reduced educational attainment. So a lower frequency of passing exams at middle school, high school, or attaining a university degree, worse performance on tests of language ability, worse reported overall health, altered alcohol intake, reduced height, reduced lung function, and a lower frequency of exercise in carriers of this expansion. And here's the results for whether individuals pass their middle school exams. You can see individuals on the left, patients or individuals that did not pass their exams have a much higher frequency of expanded alleles at this locus than people that do. So when we come to assessing the significance of a mutation like this, the two things we really want to know are how frequent is it in the population and what's its impact on phenotype. So to assess population prevalence, we went back to methylation profiles because these give us the most robust estimates of who carries these expanded alleles. And we found 38 individuals out of about 33,000 assayed, giving us an estimated prevalence of about 1 in 860. And this is surprisingly common. Just to put that in context, the Fragile X expansion is estimated to be about 1 in 5,000, whereas other important recurrent CNVs like deletion 22q11 and others have estimated prevalence of about 1 in 2,500 to 1 in 6,000. We then assessed its relative phenotypic impact on educational function versus 6 different recurrent deletion duplication disorders. So we went back to the same 168,000 UKB samples, and here I've ordered those by their effect on educational attainment. If we superimpose the effects of the AFF3 expansion here at the top, you can see its effect size is similar to that of deletions of 16p11.2 or deletions of 15q13, so other clinically significant CNVs. So we then wanted to assess its effect on rare disease. Here we turn to the UK 100,000 Genomes Project, and this had recruited almost 8,000 probands with a primary diagnosis of intellectual disability. 19% of those already had an identified pathogenic mutation. So we then genotyped the AFF3 repeat in about 6,500 probands that were unsolved despite undergoing genome sequencing, and compared this to about 9,000 neurologically normal controls. We see no difference in allele frequencies for AFF3 repeats in the normal size range. But when we look at the repeats that are the very largest size, we see an almost 4-fold enrichment in cases versus controls, which is highly significant. And if we then look at the 1,500 probands that are solved, that is, they have another pathogenic mutation elsewhere in their genome. None of these carry this repeat expansion. So this gives us very strong evidence, and we estimate that these AFF3 expansions are causal in at least 0.3% of patients with intellectual disability. This could well be an underestimate due to the difficulties of accurately assessing the size of this repeat from Illumina data. We then recruited 2 of these trios and performed long-read genome sequencing. So PacBio HiFi sequencing confirmed the presence of a highly methylated heterozygous expansion of more than 800 copies in the proband. And then looking at the parents, this was inherited, the mother carried what appears to be pre-mutation, so largely unmethylated allele of about 130 copies. This was a de novo expansion occurring in a proband from a pre-mutation in the mother. In the second trio, the proband also carried a highly methylated expansion of about 150 copies. But surprisingly, we observed here the father carried an even larger and also more highly methylated allele of about 400 repeats. And we confirmed with the clinician that there was no evidence of neurological deficits or other phenotypic anomalies in this father. Therefore, we can say that AFF3 expansions sometimes show incomplete penetrance and are compatible with normal intellectual function in some cases. Now, just to sort of wrap this up, when we were looking at this study and looking at the AFF3 gene, we looked in the GWAS catalog and found that there are actually a lot of annotated GWAS SNPs for educational function at the AFF3 gene. So just showing those here. And we wondered, could there be a connection between these SNPs found by prior GWAS and the repeat expansion? And this really touches on this issue of what's been termed missing heritability. So really, we had 2 questions. Can tandem repeat expansions be tagged by flanking SNPs? And if so, might these GWAS signals that have been found at AFF3 actually be caused by SNPs that are tagging the repeat expansion? So to test this, we went back to the UK Biobank and we performed association analysis of SNPs in and around the AFF3 locus with presence or absence of the expansion. And indeed, we found many SNPs that were very good tags for if you have the expansion. That's shown here. So we get p-values up to 10 to the minus 140. And here's the underlying data for this top hit. So this is a SNP that's present at about 20% allele frequency in the normal population, but occurs in 80% of AFF3 expansion carriers. And then when we join these with these prior GWAS SNPs for educational attainment and cognitive function, we can see the same SNPs that are the best tags for AFF3 expansions are the same ones reported in these GWAS. So what we think is happening here is that these GWAS have really found SNPs that are occurring on founder haplotypes that are enriched for the expanded alleles. This gives us further evidence for the role of AFF3 expansions in neurodevelopment, but more importantly indicates that a subset of GWAS signals are potentially driven in some cases by underlying repeat expansions, and the SNPs are really just a tag. So hopefully I've convinced you that AFF3 expansions are a significant and common cause of neurodevelopmental disorders. We would suggest that clinical testing for this is warranted in individuals with undiagnosed intellectual disability. We estimate its prevalence is about 5 times higher than the repeat that causes Fragile X syndrome. But importantly, these sometimes show incomplete penetrance, which complicates counseling. And then finally, our data suggests that some GWAS signals are potentially explained by underlying repeat expansions that touches on this issue of missing heritability. Now, this study is really just a start. We've obviously found and characterized this to some extent, but there's a lot of unknowns here. We don't know if there is a syndrome associated with AFF3 expansions. We don't even know what the minimum size threshold that might cause pathology is. And we don't know, for example, might unmethylated pre-mutations also have a phenotype like is seen in Fragile X. So to try and address this, we're interested in collecting a cohort of these patients. If anyone out there finds more of these expansions and you're interested to collaborate, please give me a call. And I'd just like to thank my lab and collaborators. Thank you.
Thanks a lot, Andy. Again, we may only have time for one question, and there's somebody rushing to the microphone already. I thought, no. Yeah, all right. Yes.
Okay, it was first, thanks. Great talk. Is there some association between the size of the repeat and the phenotype, or the probability to have a phenotype? Anything quantitative?
It's a little hard to hear. You said, is there an association between size and phenotype? Is that right? Yeah.
Number of repeats.
So it's a little hard to say. So one of the issues is when you're trying to size these, especially GCC-rich repeats, you really just get an estimate from Illumina genome sequencing. It's hard to say exactly how big they are. One thing I didn't point out, like when we PacBio sequence them, they're 500, 800 copies, but Illumina's saying they're 70 or 80 copies. So we really can't distinguish very well the size from the Illumina data. One thing I can say is we did see associations sort of across the different stringencies of expansions we use, but honestly, we don't know at this stage. It really needs long-read sequencing to size them accurately.
Wonderful. Unfortunately, we have to move on, but I encourage you to come to the front and discuss with Andy right after this session. So we'll give one more round of applause for Andy and move to the next speaker.
So our next speaker is an early career award candidate. Actually, he's a Hurley career award candidate because he's in the first year's PhD. And Kilian Solomon will speak about in silico prioritization of open chromatin sequences.